All right, this is the third of the recordings, and this one's maybe a little tougher in the sense, we're going to talk about brain parts. So we'll go through the brain and how it developed, and I do want you to be able to maybe even recognize it on a drawing, a diagram. And so I'm not a big diagram kind of guy, but this might be a diagram you want to know. So we have, don't worry about the diagrams on the left. What we're going to look at is the parts of the brain and where they are, and we have pictures on each one. But we have three kind of levels: forebrain, midbrain, and hindbrain. And as they develop differently. And you can see some develop from two parts. And this stuff I'm not overly concerned about. But I do want you to know the parts. And we're going to go through each one of these to make sure that you understand what it's kind of controlling. And so we'll start with the hindbrain in the very, very back. So we have the medulla, or the medulla oblongata, down here. And this is part of the brain stem along with the pons. And that's what hooks onto the spinal cord. And so this is in control of autonomic vital functions. Autonomic means automatic, things that you don't think about, like heart rate, blood pressure, breathing, things like that. And so this is why you hear people are brain dead but still alive, because maybe the upper portion of the brain has been destroyed, where there's no higher thought process yet. The brain stem is still functioning and causing breathing and heart rate and things like that. It also controls the mouth, soft palate, larynx for speech. So damage to this can cause the inability to speak. And also controls many involuntary reflexes that we don't think about, like swallowing and sneezing and actually vomiting, is all coming from this very lower portion of the brainstem, so the medulla oblongata. Now that works with the pons, and together they kind of coordinate signals. The pons serves as a coordination center for signals, and we have a right and a left hemisphere. And this kind of decides where that information is supposed to go for sound localization, eye movement, controls sleep. It controls making faces, so a lot of the facial muscles. These two together, that brainstem, is your very basic structures. All right, the next one is the cerebellum. It's not part of the brainstem, but it is part of the hindbrain. And this guy's cool. It coordinates all our sensations and responses and muscles, enabling most of our voluntary movements. It coordinates with the inner ear for balance. And this is the one that, we hear a muscle memory. And this is really kind of allows you to do things you don't think about. And so I want to bring up some examples. One is, today, most of you, and it's hard to tell, if you drive a stick shift versus an automatic. Most cars now are automatic, and people are scared of stick shifts because it takes some coordination. To learn to drive one, you have to work the two pedals along with the brake. But you have a clutch, you have a gear shift. And so what typically happens when people first get in, they jerk the car, to get that coordination where you're doing it smoothly, takes a while. Once you've driven one, and I have a stick, we have both actually, but I prefer a stick because I feel I have more control, and you don't really think about it anymore. So we'll give you another example for those of you that don't drive a stick. I'm assuming you walk upstairs, and maybe you're lazy and just always take the elevator. But stairs are designed a certain way, and that's because of our cerebellum. You don't really think about this, but there is a distance from where you are to where you want to be. And in this country, when you build stairs, all those stairs have to be even. You don't really think about walking upstairs. What you will do when you come up to stairs is you tend to glance down and look at that first stair, lift your foot, put it on there. After that, you don't really pay attention. That is because of your cerebellum. It's doing all the balancing. And that is why it's important in this country. We make those stairs are always a certain thickness. Now, there is a range how high a step can be in California. And what you do then is you calculate your distance from the bottom to the top, and then you divide that into how many stairs you're going to have, and it has to fall within a certain range. The stairs in my house are very narrow and they don't fit code. My house was built in 1911. So you just got to be more careful. You know, I always warn people, and everybody's spent any time in my house going up and down the stairs have fallen once. You learn. Yeah, you don't really think about it, but you have had that sensation where you're not paying attention, where you get to the top and you take that one more step and the step's not there and kind of fall, or you get to the bottom and you're on the ground, but you're trying for another step, so your knees kind of buckle a little bit. And that's due to the perception that you haven't stopped, but your cerebellum is letting you do all that. My friend, Tim Ravel, his dad was an engineer, and he built a second floor on his house. And he's a smart guy, and he calculated his steps and did all that and built the stairs. What he didn't take into account for was the 3/4 inch plywood that he had his flooring up above, plus the flooring itself. So that last stair was over an inch taller than the rest of it. So the city inspector came out and said, oh, this can't do. You got to tear all your stairs down, put them back up, which is very costly, time consuming. So his dad just removed the flooring and flattened it enough for it to pass inspection and put it back on. And so if you go to Tim's dad's old house, you would have to, as you walk the stairs, you had to realize that that last step was an extra, or if you're coming down, realize when you step down on that first step, it's bigger, or you're going to end up on the bottom. All right. The midbrain, and this is connecting the hindbrain to the forebrain. So the midbrain is a small little unit here right above the pons, and this is really kind of situated to move the eyes and localize sounds, again, try to where sounds are coming from, and sets basic brain states, alertness, drives, goals. So when you're starting to phase out, you can your midbrain is shutting down. Hopefully not during these lectures, but it could be. All right, and then the forebrain is the cerebrum, and that's the bigger part up front. That's where all the higher thinking happens, reasoning, language processing. That's where all our visual stuff and sounds and taste and smell, all of those things come into play in the cerebrum. And the cerebrum in us is very convoluted. That convolution allows for more space, and there's an article I want you to read about that in our article readings, about why our brain really can't get any better than it is. We're pretty much maxed out. And so we want to read that Scientific American, and we'll talk about that next week. All right, and they've done this, and they've done some mapping of this. And so I want to talk about this just a little bit. So we kind of know where things are happening through surgeries and damage. We got 4 major lobes. We got the frontal lobe and the parietal lobe. We've got the temporal lobes on the side and the occipital lobe in the back. And so here's the vision. You know, we've got up front is associations. We've got speech. Here on the sides, we got smell. We got hearing right around the ears. That makes sense. And everybody's areas are pretty much the same, except for, you know, somebody's speech area might be larger or smaller than somebody else's, and all same with taste. But the general areas are pretty much the same. And then the 2 hemispheres are actually maintained by what's called a corpus callosum, and that's this piece right here. And it kind of shifts information back and forth, and is formed when you are in the uterus of your mother. And one thing I want to talk about that they had experiments, I guess that's what they're called, surgeries, which I'm not sure are all that ethical now. But people with schizophrenia, they figured that the right and left brain were fighting each other. And so what they did is they went in and they thought, well, we'll just cut the corpus callosum and that'll solve all their problems. We realized that did not do the job. And so what it ended up doing, though, was kind of cool in that we found out a lot about the brain. And it turned out there was an experiment where they did that, and they handed somebody a set of keys and they put it in their right hand. And they asked them, and I don't know the right or left, I don't know if I have that right, but they put it in one of the hands, and they said, what is this called? And they said, well, this is a key. And what do you do with it? And they're like, well, I'm not really sure. And so they took the keys back, and then a little bit later, they put the keys in the left hand. And they asked the person, what are these called? And they said, oh, I don't really know. What do they do? Well, you can use them to unlock locks. So it was interesting how the brain has that information stored, but they had no access to it. And the assumption is that corpus callosum. So kind of cool experiment. And there's some brain mapping areas of the brain. There's another article. So there's really two bigger articles I want you to read, and that one is on National Geographic on the brain. So, all right. And then there's some, strange stuff they've done, cerebral cortex mapping, where they take a slice of it and they, poke and prod and figure out where things are. And then you get this, drawing done by somebody on drugs that show you all the different places where those that information is coming to in the brain. So we have this mapping, and then this is the idea. Here's one that's this a somatosensory cortex area that they've mapped, and you can see this is the drawing highlighted about sensitivity. So you can see the hands really big, the foot's really big, the lips are really big. And these are areas that are more sensitive than to some of the others. And then just to say it can be done on other animals, here's an anotypical proportion. This is a mole, a star-nosed mole. And there's a little video of them, moving around. And you can see them talk about their sensory organ here. There's their nose. But again, the sensory on the mapping on their brain shows that lots and lots of it is dedicated to the nose and obviously the front claws where there's feeling. These things are almost blind. But anyway, so this little video just showing you what these moles look like, and they're moving around talking about that a little bit. All right, and then we have the basal ganglia. It's these bluish areas here, and these are really kind of where the signal sends out for the motor function. So these are the ones that are sending out signals to tell you how to move and things like that. So that would be these basal ganglia here in the back. And then we got some others in the forebrain. We have the thalamus, which is the prime processing center for sensory information and sleep. We have the hypothalamus that controls body temperature, blood pressure, your pH of your blood, things like that. And then we have the pineal gland, this little tiny gland, which is actually used for our circadian rhythms, our internal clocks. And so all of this comes together to kind of get us thinking. And then last and not least on this section, this brain section, is we have two more parts, the hippocampus and then the amygdala. And these are kind of the memory centers. The hippocampus is really used for categorizing things and putting it away. The prefrontal cortex is where a lot of the information first comes in, and then it goes to the hippocampus, and that's kind of your short-term memory. Prefrontal cortex, you're getting this information, and that's how you can kind of hold on to stuff into the hippocampus. But it really takes some associations to get that to stick. You have to decide what's important. And then the amygdala is where our emotional responses are. And what they found is for long-term memory, you got to care about it. You've got to want to do it. You've got to work at it. You got to repetition, finding ways to associate it with something else. And actually, and I hate to say it, one of the most successful ways, they said, is, but people remember, is to make it painful. Now, it doesn't have to be physical pain, but if you have to work at it hard, they say that then you have a tendency to remember it versus just kind of trying to look at something. So the last minute cramming, you might do okay and remember some stuff. You lose a lot of it within the first 30 seconds or so. And people have done well that, but if you really want to get to know something, you got to study it. And they say it's got to be partially painful. And I've had teachers I teach with it's scary for me, but they say that we don't teach anything that's on Google because they can just look it up. Well, reality is, yeah, you can look it up, but the chances of remembering it are very, very small. And so you have to look it up again and again. Now, if you look it up enough, I guess you'll remember it. So there is benefit in trying to build a Google library in your brain versus on your computer when your power goes out and you're screwed. So we'll end the lecture. Stop the share. Stop the recording.